In the last section, I pointed out that in the cortex there are characteristic structures on different levels of detail. A structure on one level is made up of lots of structures on the next, more detailed level. On each level, there's lots of the one type of structure, and there's a general similarity between the different structures of the one type. But when you look more closely, each structure is a bit different from every other structure. All pyramidal neurons are generally similar, for example, sufficiently similar to identify the category of pyramidal neurons. But when you look closely, every neuron is different in some way, and in particular in what it's connected to. We've already talked about the more detailed levels of the cortex, the pyramidal neurons and their substructures, earlier in the course. So in the next sections, we're going to look at the two higher levels. And we're going to start with cortical columns. Now earlier I mentioned the separation of the cortex into layers, with layering visible everywhere in every cortical area. And as we'll get to later, there are some differences in the layering between different parts of the cortex. But in the reference view, 6 layers are counted, even though sometimes a layer may be subdivided and sometimes a layer seems to be missing. Layer 1 on the outside, closest to the skull, is mainly axons, so it looks white in this Nissl stain. Layer 2 typically contains lots of relatively small pyramidal neurons. Layer 3 contains pyramidal neurons that increase in size as you go from the boundary with layer 2 down to the boundary with layer 4. Layer 4 contains very small spiny stellate neurons, very large numbers of them in the areas that process the initial inputs from the senses. The pyramidal neurons in layer 5 are large, while the pyramidal neurons in layer 6 vary in size with no clear pattern across the layer. Now, cortex sections can be stained in different ways to reveal different aspects of its structure. On the left, you've got the Golgi stain that reveals complete neurons, including soma, dendrites, and axons, but it only actually reveals about 2% of the neurons. In the middle Nissl stain, it only reveals somas, but a high proportion of the somas are generally visible. And on the right, the stain reveals just axons. So there's a number of structural considerations revealed by these different approaches. The stain on the left reveals that pyramidal neurons tend to be oriented with their apical dendrites perpendicular to the plane of the cortex, and their basal dendrites roughly parallel to the layering, and often confined to the same layer as the soma. The middle stain reveals the varying soma sizes. For example, the increasing size going deeper into layer 3. The right-hand stain reveals a pronounced connectivity vertically across the layers, but also a pronounced lateral connectivity within some layers. Another aspect of cortical layering is a pronounced pattern of connectivity, both locally and long distance. Layer 4 receives a high volume of inputs from the thalamus. Layers 2 and 3 have a lot of connectivity with other cortical areas, especially the cortical areas associated with the hippocampus, as we'll describe later. Layer 5 provides lots of outputs to the basal ganglia and also to the cerebellum. Layer 6 provides outputs to the thalamus and to other cortical areas, especially distant cortical areas. Then there's also a pronounced pattern of vertical connectivity. Layer 4 targets layers 2 and 3, and layers 2 and 3 target layers 5 and 6. Although, in fact, some layer 4 outputs go direct to layer 6. So the primary local flow of connectivity is vertical. External inputs arrive in layer 4. Layer 4 targets layers 2, 3. Layers 2, 3 targets layers 5, 6. And layers 5, 6 provide the external outputs. This vertical flow of connectivity reflects a pronounced column structure perpendicular to the layers. On the smaller scale, in embryonic development, microcolumns of about 100 neurons grow from one or two precursor cells. One microcolumn contains all cell types, and microcolumns have been observed in all mammal species. In humans, these microcolumns vary in diameter from maybe 35 to 60 microns. But there are also columns that are formed by groups of 50 to 80 microcolumns that all have the same inputs and lots of short-range horizontal connections. And these columns vary between 300 and 500 microns in diameter. Columns of about this same size again exist in all mammal species, even though the brains vary in size by a factor of 1,000. So in the human cortex, there are several million of these columns, fewer in smaller brains. Now, something very interesting about columns was revealed by studies of a cortical area called TE in the cortex of the macaque monkey. This area processes visual information. Information derived from the eyes enters the primary visual cortex area V1, then goes from V1 to an area called V2, then to V4, then through an area called TEO to reach TE. And it's found that damage to area TE results in severe deficits in learning tasks that need visual recognition of objects. And neurons in TE are activated in complex situations that combine orientation, size, color, and texture information about visual objects. These images represent the receptive fields of different TE pyramidal neurons. In other words, what these neurons are detecting within visual input. And these representations were obtained by probing individual neurons in the monkey cortex and monitoring their activity in response to a range of different visual objects. The representations are the result of processing to identify the kind of common denominator in the responses of different individual neurons to the range of objects. Now, one obvious comment is that these receptive fields don't correspond with categories of visual objects, even though TE is important for identifying such categories. If pyramidal neurons in nearby locations in the cortex are probed, it's found that all the cells in a column with a diameter of about 0.4 millimeters have similar, although not identical, receptive fields. But when you cross a boundary, there's an abrupt change to a completely different and unrelated receptive field. So the cortical area is divided up into columns on the basis of different receptive fields. Within a column, all the pyramidal neurons have similar receptive fields, so these images could also be regarded as the receptive field of the column, but there are no obvious relationships between the fields of different columns, even for adjacent columns. It's also interesting to look at which columns are active in response to different visual objects. The large picture on the left shows where there was activity on the surface of the monkey cortex in response to the three images on the right: a cat, the head of a cat, and the silhouette of a cat head. Underneath each image is a different colored line, and the cortical columns active in response to each image are circled by a line of the corresponding color. So the active cortical columns in response to the complete cat are circled in red, the areas active in response to the cat head in blue, and for the silhouette in yellow. You can see that many columns are active in response to one object, and if the objects are visually similar, some of the same columns are activated. Similarly for fire extinguisher images, and again, there's a different colored line under each of the four images on the right, and the active cortical columns in response to each image are circled on the larger image on the left. But note that in this case, there's a receptive field, circled in green, that's detected in a component of the full visual object, but not in the complete object. Another significant observation is that when the brain sees different types of object, sometimes the same column detects its receptive field, perhaps especially if there's some partial degree of visual similarity. And these 8 different visual objects were shown to a monkey, and activity in some of the monkey cortex was monitored. And this shows 2 monitored regions within area TE, looking down on the layers. Each region is about 4 millimeters by 2 millimeters, so a column corresponds with an area about 0.5 millimeter by 0.5 millimeter, and I've indicated the rough area on the right. This shows the regions that were active in response to each different object. For example, those are the columns activated in response to object 1. These columns were active in response to object 2. These columns in response to object 3. So different groups of columns are active in response to different objects. But the group of columns active in response to one object may partially overlap with the groups active in response to one or more other objects. For example, the columns active in response to objects 1, 2, and 3 partially overlap. One of the columns active in response to object 5 coincides with a column active in response to object 6. So columns don't correspond with categories of visual object, but the set of columns responding to one object is different from, but can partially overlap with, the set activated in response to a different type of object. Those types of receptive fields we've been discussing are defined by pyramidal neurons. But there's also significant numbers of inhibitory interneurons in the cortex. And the structure of these inhibitory neurons, connectivity and so forth, correlates with columns. This shows a cross-section of the cortex with 6 layers, and the diameter of a typical column is illustrated. And a number of different types of interneuron are shown. The dendrite is drawn in black and the axon in grey. In many cases, the axons and dendrites are largely confined within one cortical column and within 1 or 2 cortical layers. A key variable for an interneuron is the location on pyramidal neurons on which they synapse. Some interneurons target dendrites a long way out from the soma. Other interneurons target dendrites close to the soma, and yet others target the actual somas. Some even target the early stages of the pyramidal axon. And these differences in position determine the type of effect an inhibitory interneuron input will have. So, inhibitory inputs add to the complexity of the integration process for synaptic inputs to a pyramidal neuron. If over a period of time excitatory inputs are scattered all over the dendritic tree, they'll actually have very little effect. To produce an output action potential, the same number of inputs must be concentrated on a small number of terminal dendritic branches and within the integration time window. But depending on its location, an inhibitive input can reduce the effect of just one terminal branch, or one whole dendrite, and an inhibitory input on the initial segment of the axon will exert a very powerful regulatory effect on the outputs of the neuron as a whole. And because a high proportion of the inhibitory interneurons' inputs and outputs are generally within one column, they are key to coordinating the activity of the column.